Hello, my name is Abhi Gopal and I'm a representative from AWAP, an IBIT group. Now I'm here to talk to you give you a little intro about our tech component, which is going to be an algae bioreactor. Now our project is basically cleaning up oil spills with algae, so it might be a little vague why we need an algae bioreactor. But since TJ Star, the day our due date, the day the presentation is there, is in May, we have to work on do this project a majority in wintertime, where it's very cold and algae doesn't really culture that well, and we can't afford to get a bunch of algae and just do it for our project. So we have to build this bioreactor, which is pretty much going to culture the algae for us, and regardless of the temperature, the weather, amount of sunlight, going to separate nutrients. And Prajit is going to talk to you about that.
Thanks, Abhi. So yeah, as he said, I'm going to explain more on basically the design brief of this tech component. What happens is we have a problem and some constraints and some risks opposed to the user. Now, let me explain to you what exactly those three things are. The problem is, as Abhi said, we have to grow these things in a short amount of time in cold weather. Now, if you know your algae, you know that it takes a long time to grow algae, and algae doesn't typically grow in the Arctic, if you know what I mean. It grows near Florida, really hot, equatorial places. So that's actually the problem we have. How to grow algae in the winter and with the limited time. Because we have to finish this algae growing stuff by January. Okay, and next we're going to talk about the constraints we have. See, the constraints here are really simple in this tech component of the algae photobioreactor. There's only three constraints we have. Everything else we can manipulate as much as we want. The time. As Abhi said, we only have until May to complete the whole project and until January to actually grow the algae so we can test on them and do whatever we need to do at that time. Another constraint is the weather. It's really cold outside and you can't grow algae in the winter. And I know this is getting a bit repetitive, but I'm just trying to make sure that you know what our design brief is. And the third one is, we have a limited amount of supplies. The photobioreactor takes a specific amount of supplies to build. It's not just, you grab those supplies, you grab those supplies, and bring them together and make a photobioreactor. That doesn't work. We have a limited amount of supplies, limited amount of money, and it just doesn't work for us. So that's the third constraint, the amount of supplies we actually can get or own right now. And the third part of this design brief is the risks involved. In my lifetime, I have not seen any killer algae before, so I'm pretty sure that there are no risks to the user of this project. Except maybe the algae could have gotten hurt, if you tear them, rip them, and I'm pretty sure we won't do that, but that's why we're taking extra precaution and we're going to handle them safely. And that's the design brief. Okay, I'm here to explain more about the resources. Basically, we'll have chipwood, water bottles, tubes, a carbon source, and heat lamps. And these supplies also solve our problems that we stated in previous sections of this video, such as all the items are cheap. They're very efficient also, so that solves our problem of the limited amount of supplies that we have. They're also easily accessible to the average person. It saves time for us because we don't have to go contact the manufacturer for specific parts and whatever. Because water bottles they're not really that complicated, if you know what I mean. And the weather aspect of the problem, we don't need to worry about that either because our heat lamps can counteract the cold weather to a certain degree. I'm not saying it'll make the world heat up in the middle of January. What I'm saying is that it'll provide some balance so that the algae can survive in the cold weather. Thanks. Next, as a follow-up to the resources, I'm going to talk about the strategy. Specifically, how I'm going to go implementing the engineering process and talking about what's my timeline, how long it's going to take to build the photobioreactor, and how long it'll take to actually measure the results. Okay, so here's our timeline, first of all. Circa January, the first week of January, to be specific, we will gather supplies for the reactor by that week. And throughout that week, we'll build it. And that ties in with my second point. Circa January, the second week, we will build the bioreactor. So by the second week, the bioreactor will be done, finished, at one of my group members' house. And then we'll wait 1 month, or 4 weeks, for the algae to culture. And we're giving the algae some ample time to culture because we want good algae, not hasty, run-of-the-mill. And for the remaining time from after we gather our results, we're just going to prepare for TJ Star by making similar videos and preparing all aspects, lit review, and all those other aspects of the IBET project. How long will it take to implement various parts of this project? 1 to 2 weeks max. And I know that seems like that's a lie, but it's true. Because our clever solutions to those constraints, it helps us. It helps us a lot because we are now endowed with the power to bypass those constraints, and now we can create the photobioreactor with ample time to go and create other parts of the bioreactor if we want, so we can make the project better. And we really don't need special tools for this, since it's not really that complicated, since we're using wood and a hot glue gun. So yeah, the most specialized tool we'll be using is a hot glue gun. And what needs to happen before we can begin to continue work? We'll need to gather supplies. And that's pretty much the most important aspect of this photobioreactor. So we can gather supplies, so we can build a reactor, and thus we can culture the algae so we can experiment on. And that's the strategy that we will use to implement the photobioreactor in our IBET project. Thanks.
Hello, this is Abhi Gopal again, and I'm here to talk to you a little more into detail about the algae bioreactor. Now here's a basic blueprint of what this is going to look like. So these are the water bottles that you've heard of, and these are actually going to be the tubes, plastic tubes that we're going to pick up from Home Depot. This is going to be a carbon dioxide source. It doesn't have to be a tank per se, but it probably is going to be. And this is just a simple wood sheet that all of these bottles are going to be mounted onto with something probably really basic like hot glue. Just so, for portability reasons, the wood plank doesn't actually do anything in the actual process itself. So I'm going to teach you a little more about the biological viewpoint of what's actually going to happen here. So first off, we're going to fill these bottles with algae. So here's little green algae. I'm just going to fill one bottle just for the example, but in reality, just know there are going to be more. Now, after that, it's going to go through two processes. All plant cells do. One is called photosynthesis, which you probably heard of, and the other is cellular respiration, which you might have heard of. Now, photosynthesis requires two main inputs or reactants. One would be water, and then you need carbon dioxide. You also need light energy, which isn't pictured here. But let's just pretend there's the sun here, or a lamp or something like that. It's going to give sunlight, or just light and heat. So what's going to happen is the light is going to go to the algae, which is going to give it the light aspect. We're also going to fill these bottles with water, which is probably going to be nitrogen rich because we're going to put plant fertilizer inside it. We also need carbon dioxide now, which is conveniently right in this tank. So since it's hooked up so tightly to it, the plant doesn't really need to try that hard to get the carbon dioxide. So it's just going to go from the tank. Let's say this blue line is the carbon dioxide. It's going to go through all of these bottles and just transfer carbon dioxide to all of the bottles. So along with the sun's rays and the carbon dioxide, we're going to have enough of the materials to pretty much do photosynthesis. Now what photosynthesis equals, it gives an output of oxygen and glucose. Now glucose is pretty much a basic sugar. It's used in cellular respiration along with oxygen. And this oxygen is kind of special actually, because out in the nature, plants actually grow slower because the oxygen is immediately lost because it's just in the open. But this is actually an airtight environment, so the oxygen is going to be trapped in to perform cellular respiration. So this oxygen, O2, plus the glucose is going to perform cellular respiration, and the output of that is going to be carbon dioxide, CO2, and it's also going to provide ATP. Now, these two things are going to play a pivotal role. Now, the CO2 is what makes it a cycle. The CO2 is going to go back and perform photosynthesis again, in addition to the CO2 in the tank. So this tank is never really going to run out, because as soon as it gets started, the source, more carbon dioxide will continuously be produced, so it will never run out of carbon dioxide. And the oxygen won't run out, because after the carbon dioxide is there, the oxygen will come back up. Now the ATP, I'm going to adjust this, please, is pretty much what we're aiming for. It's a source of energy, adenine triphosphate. So it's an energy carrier, basic energy carrier that all cells use. And what it's going to do is it fuels all of the cell's functions. In this case, we're going to make a lot more ATP, since the plant doesn't have to wait to get oxygen or CO2 the natural way, since they're already so close to the plant. Like, in reality, this is probably only a few feet away. So that's what speeds this whole process up. Now, this ATP is going to speed up the functions, which is going to speed up photosynthesis, and it's going to grow at a much faster rate and reach the maturation rate, age a lot faster. So it's pretty much going to become mature a lot faster, in simpler terms, and grow a lot more. Now, this is going to help us, because after we're done, we can just take the algae out, and we'll have a lot more algae than we started with over short amounts of time. We don't have to wait for the materials to go in, like the CO2. We don't need to wait for that to go back, and we don't need to wait for like the oxygen to get in. And we also don't really need that much heat or light, which is usually to take in winter. We're doing a majority of this project, as you probably know, in winter time, so there isn't much like heat going on, which is why we're probably going to have a non-pictured like here, but a lamp to heat this up, and the light and heat are going to be trapped in through these plastic water bottles, and are just going to stay within the system, so they won't overheat, because there'll be like enough ventilation, but they're going to keep the plants warm. So this is pretty much how the algae bioreactor works in a simple viewpoint, scientific viewpoint. If you have any questions, you can email at awapproject@gmail.com. So this is actually really easy to do. A lot of people have done this before. This isn't actually anything new project, like everything that's going to happen there. So I hope you enjoyed this presentation. If you have any questions, email us. I'll put the email address in the bottom box, and a source that we got some information from. It's in Instructables.com. If you want to build your own algae bioreactor, or just look more deeply into it, you can click the link at the bottom description box. That's pretty much it. Thanks for watching. I hope you enjoyed. Hope we didn't bore you too much. Thanks.